Hello friends. So today we are continuing our discussion on hemostasis in pathology. So now we are talking about second component of coagulation. That is hemostasis. Second component of hemostasis, that is coagulation cascade. We have already discussed about platelet in previous video. So coagulation cascade is a series of amplifying enzymatic reaction that leads to a deposition of insoluble fibrin clot. As discussed later, the dependency of clot formation on various factors differs in the laboratory test tube and in blood vessel in vivo. However, clotting in vitro and in vivo both follow the same general principle as follows. So you can see here coagulation cascade in laboratory and in vivo. This is clotting in laboratory and this is clotting in vivo in human body. Clotting is initiated in laboratory by the adding phospholipid, calcium, and either negatively charged surfaces such as glass bead. It will result in intrinsic pathway, or we can start uh, clotting in by adding tissue factors, which results in extrinsic, which is known as extrinsic pathway. So, clotting in laboratory follows two type of mechanism: intrinsic pathway and extrinsic pathway. So, recall from physiology, what are what happens in intrinsic pathway? 2L will convert in 2L-A Hagman factor in presence of negative charge surface like glass bead. 2L-A will catalyze 11 conversion of 11 into 11A and 9 into 9A with 8A, it will convert it to 10 into 10A and finally prothrombin. This is 2 second into thrombin. This is clotting factor second, prothrombin. So it will convert in thrombin and thrombin will catalyze fibrinogen conversion into fibrin clot. In extrinsic pathway, it's the uh, clotting factor 7 will cause tissue factor uh, uh, clotting tissue factor will cause activation of factor 7 into factor 7, and factor 7a activated factor 7 will catalyze a conversion of uh, 10 into activated 10a, which will again catalyze formation of prothrombin into thrombin and finally ultimately fibrinogen into fibrin clot. This was about clotting in laboratory. These are discussed in detail in physiology. Recall from physiology. So what happens in clotting in vivo? It is the same basic principle: tissue factor. It will come first on the but here it is little bit different. It will 9 will be converted into 9A. Rest all are almost same. And thrombin sets positive feedback mechanism in for 5A. You can say 8 for catalyzing. In vivo, tissue factor is major initiator of coagulation. You can see here, which is amplified by feedback loops involving thrombin, the dotted line. Red polypeptides are inactive factor. The dark green polypeptides are active factors, and light uh, light green polypeptide correspond to cofactors. This is light green are cofactor. Reaction accelerator. Factors marked with asterisk like 10, 9, 10, 9, 7. These are vitamin K dependent, as are protein C and S. So which clotting factor are depends on vitamin K factor 2, 7, 9, 10 and protein C and S, not given, not depicted here. Warfarin act as anticoagulant by inhibiting the gamma carboxylation of the vitamin K dependent coagulation factors. Vitamin K is an essential cofactor for synthesis of all these vitamin K dependent clotting factors. So let's discuss. The cascade of reaction in the pathways can be linked to a dance in which coagulation factors are passed from the one partner to the next. Each reaction steps involve an enzyme or an activated coagulation factor or substrate, an inactive proenzyme form of coagulation factor, and a cofactor or reaction accelerator. So it will The reaction step involves enzyme, substrate, and cofactor. These components are assembled on negatively charged phospholipid surface, which is provided by activated platelet. Assembly of reaction complex also depends on calcium, which bind to gamma carboxylated glutamic acid residues that are present in factor 2, 7, 19. The enzymatic reaction that produces gamma carboxylated glutamic acid use vitamin K as a cofactor and are antagonized by drugs such as coumadin used as an anticoagulant. Coumadin is used as an anticoagulant. You can see it. Schematic illustration of conversion of factor 10 into 10A, which in turn 10A, which in turn converts factor 2 into factor 2A, thrombin. The initial reaction complex consists of proteolytic enzyme 9A. You can see this is proteolytic enzyme 9A. And a substrate, 10 is a substrate, factors 10. And reaction accelerator is factor 8A. You can see this green color, reaction accelerator. All assembled on platelet phospholipid surface. This is platelet phospholipid surface. This little yellow color, little yellow brown color. This box type of thing is platelet phospholipid surface. Calcium ion holds the assembled component together. You can see here, 9 and it was 9 and 10. 2, 7, 19 will require calcium. These gamma carboxylated residues uh, are held by calcium. You can see here again. Uh, you can say assembly of reaction complex depends on calcium. Calcium binds to gamma carboxylated glutamic acid, and this gamma carboxylated glutamic acid residue is present in 2719. So calcium hold the assembled component together and are essential for reaction. Calcium is itself coagulation factor number 4. Remember from physiology. Activated factor 10A becomes the protease for the second adjacent complex in the coagulation cascade, converting prothrombin substrate 2 into thrombin 2A using factor 5A as a reaction accelerator. 5A as a reaction accelerator. You can see this blue color cylinder, this red pink color cylinder is factor 5A. This is the reaction accelerator. Factor 10A will convert factor 2 into activated 2A. Again, calcium is required for binding. So, based on assays performed in clinical laboratories, the coagulation cascade can be divided into the extrinsic and intrinsic pathway. So, we have already seen in picture. The prothrombin time accesses the function of protein in the extrinsic pathway. Factors 7, 10, 5, 2. 2 is prothrombin and fibrinogen. In brief, tissue factor, phospholipid and calcium are added to plasma and the time for a fibrin clot to form is recorded. Normal is 12 seconds according to guidon. Normal is 12 seconds. In prothrombin time assay, it accesses the extrinsic pathway function. Partial thromboplastin time PTT assesses the screens of function of protein in the intrinsic pathway. Factor 12, 11, 9, 8, 10, 5, 2 and fibrinogen. In this assay, clotting of the plasma is initiated by the addition of negatively charged particle. Example, ground glass that activates factor 12 Hagman factor together with phospholipid and calcium, and the time to fibrin clot formation is recorded. So PETT, this is little bit lengthy, so it will result in length. So it will access little bit lengthy pathway, that is intrinsic pathway. Prothrombin time, this is little bit shorter name. So it will access the function of extrinsic pathway, which is little bit shorter, which involve little shorter step. So this may be the way of remembering. Although the PT and PTT assays are of great utility in evaluating coagulation factor function in patient, they do not recapitulate the event that lead to coagulation in vivo. This point is most clearly made by considering the clinical effect of deficiency of various coagulation factors. Deficiency of factor 5, 7, 10, 9, 10 are associated with moderate to severe bleeding disorder, and prothrombin deficiency is likely incompatible with life. So if person has prothrombin deficiency, then he will not survive. In contrast, factor 11 deficiency is only associated with mild bleeding An individual with factor 12 deficiency do not bleed and in fact may be susceptible to thrombosis. This is little bit paradoxical. Factor 12 deficiency will not cause bleeding. In fact, it will cause thrombosis. It seems that it will do opposite than other. But why is this so? We will discuss later. By contrast, there is evidence from experimental models suggesting that in some circumstances, factor 12 may contribute to thrombosis. This paradoxical finding may stem from the involvement of factor 12 in the several pathways, including the pro-inflammatory bradykinin pathway as well as fibrinolysis. Fibrinolysis we'll discuss later. Based on the effect of various factor deficiency in human, it is believed that in vivo, factor 7A or tissue factor complex is the most important activator of factor 9 and that factor 9A, factor 8A complex is the most important activator of factor 10. So 9A, 8A complex is most important activator of factor 10. So we have seen in previous picture. The mild bleeding tendency seen in patients with factor 11 deficiency is likely explained by the ability of thrombin to activate factor 11 as well as factor 5 and 8, a feedback mechanism that amplifies the coagulation cascade. Among the coagulation factors, thrombin is the most important, and because its various enzymatic activities control diverse aspects of hemostasis and link clotting to inflammation and repair. Among thrombin's most important activities are the following. So now we are going to discuss about activities, what importance of thrombin. Thrombin is very important not only for hemostasis but also for tissue repair, inflammation. We will see now. So its first function, conversion of fibrinogen into cross-linked fibrin. Thrombin directly converts soluble fibrinogen into the fibrin monomer that polymerizes into an insoluble fibril and also amplifies the coagulation process not only by activating factor 11 but also by activating two critical cofactors, factor 5 and say 8. It also stabilizes the secondary hemostatic clock by activating factor 13, which covalently crossing fibrin. Second function, platelet activation. Thrombin is a protein inducer of platelet activation and aggregation through its ability to activate PAR1, thereby linking platelet function to coagulation. So we have discussed this about in previous video. What is full form of PAR? Protease activated receptor 1. It is G protein coupled receptor, and thrombin is protein inducer of platelet activation and aggregation. We have seen in previous video. Remember. Third, pro-inflammatory effects. PAR also expressed on inflammatory cells, endothelium and other cell types, and activation of these receptors by thrombin is believed to mediate pro-inflammatory effects that contribute to tissue repair and angiogenesis. You can see here a role of thrombin in hemostasis and cellular activation. Thrombin plays critical role in generating cross-linked fibrin by cleaving fibrinogen into fibrin and activating factor 13, as well as activating several other coagulation factors. You can see here fibrin. Through protease activated receptor PARs, thrombin also modulates several cellular activities. It directly induces platelet aggregation. You can see it directly induces platelet aggregation and thromboxane A2 production. Thromboxane A2 production and activates endothelial cell, which respond by expressing adhesion molecule and cytokine mediators. Platelet-derived growth factor. Thrombin also directly activates leukocyte, extracellular matrix, platelet-derived growth factor for additional anticoagulant activities mediated by thrombin. You can see that thrombin causes neutrophil addition. It will cause monocyte activation. It will cause release of platelet-derived growth factor, which act on smooth muscle cell, lymphocyte activation, endothelium activation. Thrombin has variety of functions. Fourth is anticoagulant effect of thrombin. Remarkably, through mechanism described later on encountering normal endothelium, thrombin changes from procoagulant to an anticoagulant. This reversal in function prevents the clot from extending beyond the site of vascular injury. So thrombin performs opposite function in normal endothelium. It has anticoagulant effect in normal endothelium, but in injured blood vessel it will cause coagulation. Factors that limit coagulation. Now we will talk about factors that limit coagulation. Once initiated, coagulation must be restricted to the site of vascular injury to prevent deleterious consequences. One limiting one limiting factor is simple dilution. Blood flowing past the site of injury washes out activated coagulation factor, which are rapidly removed by the liver. Second is the requirement for negatively charged phospholipids, which are mentioned are mainly provided by platelets that have been activated by contact with intraendothelial matrix at the site of vascular injury. However, the most important counter-regulatory mechanisms that involve factors that are expressed by endothelium adjacent to the site of injury. So, activation of the coagulation cascade also sets into motion a fibrinolytic cascade that limits the size of clot and contributes to its later dissolution. Fibrinolysis is largely accomplished through the enzymatic activities of the plasmin, which breaks down the fibrin and interferes with its polymerization. An elevated level of breakdown product of fibrinogen, often called fibrin split products, most notably fibrin-derived D-dimer, are useful clinical marker of several prothrombic states. Plasmin is generated by enzymatic catabolism of the inactive circulating precursor plasminogen, either by a factor 12 dependent pathway, possibly explaining the association of factor 12 deficiency and thrombosis, or by plasminogen activator. So, how factor 12 deficiency causes thrombosis? You can see here. Factor 12 help in catabolism of plasminogen into plasmin. Factor 12 in factor 12 dependent pathway. So, in factor 12 deficiency, uh, plasminogen will not convert into plasmin, and plasminogen will not convert into plasmin, and there will be excess formation of fibrinogen thrombus. Most important plasminogen activator is the TPA, tissue plasminogen activator. It is synthesized principally by endothelium and is most active when bound to fibrin. This characteristic makes TPA a useful therapeutic agent since its fibrinolytic activity is largely confined to the sites of recent thrombosis. Once activated, plasmin is in turn tightly controlled by counter-regulatory factors such as alpha-2 plasmin inhibitor, a plasma protein that binds and rapidly inhibits free plasmin. So let's see the same text in the picture. This is little bit complicated. Let's see the picture. What this text want to say. So you can see here. Endothelium will produce tissue plasminogen activator TPA and urokinase, which helps in fibrin degradation of fibrin clot. These are our this is fibrin clot, platelet, plasmin. Plasmin will cause fibrin degradation. Thrombin will act on endothelium. Alpha 2 antiplasmin. This free plasmin is bound to anti-alpha 2 antiplasmin and limits and inhibits free plasmin. Let's see the picture. Now let's see the text again. Let's read the text. We have seen the picture. Now we will have general idea what are the things. Keep in mind this picture and let's again see the picture. This is a little bit complicated and difficult. So let's see it again so that it will be we'll have strong memory about the text. So activation of the coagulation cascade also sets into motion a fibrinolytic cascade that limits the size of clot and contributes to its later dissolution. So coagulation cascade sets fibrinolytic cascade means fibrin the lysis of fibrinolytic means fibrinolysis is largely accomplished through the enzymatic activity of plasmin, which breaks down fibrin and interferes with its polymerization. You can see a picture, it is clear. Plasmin helps in breakdown of fibrin and interferes with its polymerization. An elevated level of breakdown product of fibrinogen, often called fibrin split products, most notably fibrin-derived D-dimer, are useful clinical marker of several thrombotic state. Plasmin is generated by enzymatic catabolism of the inactive circulating precursor plasminogen, either by factor tubal dependent pathway or by plasminogen activator. You can see how plasmin is generated. You can see from here figure tissue plasminogen activator. You can see most important plasminogen activator is TPA, synthesized by endothelium, and is most active when bound to fibrin. You can see here, it is produced by endothelium and most active when bound to fibrin. Tissue plasminogen activator. This characteristic makes TPA a useful therapeutic agent. Once activated, plasmin is in turn tightly controlled by counter-regulating factors such as alpha-2 plasmin inhibitor, or plasma protein that binds and inhibits free plasmin. So free plasmin will be inhibited by alpha-2 antiplasmin. So we have seen the clotting factor. Now let's discuss the key concept. What are the summary of all these things? Coagulation occurs via the sequential enzymatic conversion of cascade of circulating and locally synthesized protein. Tissue factor elaborated at the site of injury is the most important initiator of the coagulation cascade in vivo. At the final stage of coagulation, thrombin converts fibrinogen into insoluble fibrin that contributes to the formation of the definitive hemostatic plug. Coagulation normally is restricted to the sites of vascular injury by limiting enzymatic activation to phospholipid surface provided by activated platelet or endothelium. Circulating and inhibitor of coagulation factors such as antithrombin 3, whose activity is augmented by heparin-like molecule. So these things we will discuss later. We will discuss about this later. These are given in later text. So let's move forward. Let's not confuse ourselves. Just remember up here. We have finished up to coagulation is normally restricted site of vascular injury. Endothelium. So we'll discuss about the role of endothelium in hemostasis. Balance between the balance between the anticoagulant and procoagulant activities of endothelium often determines whether clot formation, propagation, or dissolution occurs. Normal endothelial cells express a multitude of factors that inhibit the procoagulant activities of